what most of you will know, of course, is that while the typical cell is a diploid cell with 2 copies of each chromosome, the cancer genome very frequently is aneuploid, that is other than a multiple of the haploid set. And indeed, if you look at tumors, especially solid tumors, most of them, almost all of them have the wrong number of chromosomes, and even more so, not only if you stop here you can see one of these karyotypes where, yeah, there's 3 copies of chromosome 14, 3 of 13, but you can look down at the bottom, you have chromosomes that have been really highly rearranged. And I admit, I was always confused by that. How do we generate that? And for me, a fundamental insight came from this work from Peter Campbell and his colleagues in 2011, when he discovered that what should have been a normal chromosome 5, by using whole genome sequencing and then reconstructing that chromosome, that actually it had been shattered into hundreds of pieces, and those pieces had been put back together in a random order, amplifying some of them and dropping out others. And he called this chromothripsis, for the shattering and religation of the chromosome. And indeed, we now recognize that some tumors, almost all of the class of tumors has a chromothriptic chromosome, and indeed there's kind of a challenge now about among those who are doing the genomics here of what is the final number, and it's somewhere between 20 and 50% of human tumors have a chromothriptic chromosome. The story of chromothripsis actually began in 1968, when this pair of investigators at the NCI discovered quite correctly that if you missegregate a chromosome, or a acentric piece of a chromosome, into what we call a micronucleus, a small encapsulated chromosome, but not part of the major nucleus, that micronucleated DNA will replicate slowly, maybe not finish, by the time the major cell gets into the next mitosis, and that it will be shattered, or in their words, pulverized in the next mitosis. And that publication was forgotten. It was forgotten for 44 years until it was rediscovered by David Pellman and his colleagues at Dana-Farber. And sure enough, they demonstrated, yes, indeed, if you segregate a chromosome, missegregate a chromosome, so it like this one here that I'm circling, and that it forms a micronucleus in the subsequent interphase, that DNA will be pulverized, will be shattered. Now, yes, we discovered that the mammalian centromere is epigenetically defined by a chromatin structure modeled by a centromeric histone H3 called SIMP-A, whose chromatin knows how to specify its own replication. And that's the epigenetic mark of centromere identity. And there's an anomaly of the Y chromosome, and we learned how to selectively inactivate just the Y centromere so that we can missegregate the Y chromosome at will. And when we do that, as you can see here, we do that, and then very next interphase, you see that it is incorporated into a micronucleus. Here's another example here. And what happens to that missegregated Y chromosome in a micronucleus? And as the cell gets to the subsequent mitosis, what you can see here using fluorescent situ hybridization to follow the Y, you can see the Y in the green just gets shattered, in this case, into at least 25 viewable pieces, big enough to be seen using this in situ hybridization approach. So indeed, segregation of a chromosome or a chromosome piece, an acentric piece, into a micronucleus will lead to its shattering in the subsequent mitosis. We then ask, well, what if there were a growth advantage in the shattered chromosome? What would that do to acquisition of that growth advantage and maintenance of that growth advantage? And for the guys in the audience, you may know, of course, that we don't have any useful genes on the Y, at least in the typical somatic cell. So we added one, a neomycin resistance gene. And then we ask, if we inactivate the centromere, send that that chromosome into a micronucleus. It's going to be shattered. And in some cases, it'll be shattered to make an acentric piece. And in other cases, it will carry the original centromere, which we can either leave off or we can selectively reactivate by the strategy we used. And so what do you get if you now add that growth advantage, select for the growth advantage, and if you look at an acentric piece, so where we do not reactivate the centromere, and what happens is, and we're going to follow the pieces of the Y using a paint for the P and Q arms. And what you see is that the fragmented chromosome, no centromere, it can only be maintained, or it can be maintained if it joins another chromosome. And here it did so. It did so again. But you get some really complex rearrangements, as you can see in the fish, more complex ones, some really weird ones. So indeed, you can, if you allow the centromere to be reactivated, you get a chromothriptic Y chromosome rearranged. But if you inactivate the centromere and keep it off, you get both intra and interchromosomal rearrangements that really mimic all of the major rearrangements found in the cancer genomes. And partnering with Peter Campbell at the Sanger, we did whole genome sequencing of about 50 of these. And what we learned very quickly was, indeed, by selecting for that growth advantage, the retention of the neomycin resistance gene, you get chromothripsis. And indeed, it's just easy to see of the pieces and the wild rearrangements that you can generate here from the piece of the Y joining now a different autosome, the chromosome 5. Okay, so, and indeed more so, what you also see, and that's going to become the theme for the remainder of the talk today, what you see is that the fragmented chromosome carrying the resistance piece can circularize. In this case, in the very simple example, it can circularize into a two-piece element with only less than a megabase in size, carrying the original gene and another piece, in this case of the Y chromosome, but ligated backwards in the wrong order. So we can get, and this is amplified. The original discovery of this kind of circular DNA came from Bob Schemke and Fred Alt in 1978, and they called it a double minute because after DNA replication, it looks as two little dots. I'll show you that in a moment. It's been renamed by my colleague, our former colleague, Paul Michel, as extracellular DNA, probably a better name. And here it is. You can see in a colorectal cancer cell line, you see two kinds of gene amplification. One, an intrachromosomal amplification called a homogeneous staining region. These regions were originally identified by Barbara McClintock now 80 years ago. But it's certainly found broadly in cancer here for an amplification of the MYC gene. And indeed, you see in the same cell, you see this intrachromosomal amplification as well as the plethora of these extrachromosomal ecDNAs, or for the old timers, double minutes. Okay, and just over the last 2 to 3 years, it's become really clear, again from the work of Paul Michel and Vineet Bafna, that the up to 50% of cancers contain such amplified ecDNA. Here's one such image from a glioblastoma cell line where you see the major chromosomes, but you see all these little dots, double dots, after DNA replications. These are the double minutes or ecDNAs, and indeed you really find them very broadly in glioblastoma, but also in multiple cancer types. So, and indeed, the recognition that ecDNA is broadly found in human cancers led the two major world funders, the NCI and the Cancer Research UK, led them in just prior to COVID in 2020 to issue 9 grand challenges of unsolved, what they proposed were unsolved questions. And one of those was gene application and extrachromosomal DNA. They proposed six primary questions. What drives ecDNA formation? How can you target cancers driven by ecDNA? How do we target the ecDNA evolution? All the, and can we eliminate ecDNA? Okay, so we set out, and a postdoc of mine, Ofer Shoshani, now just a month ago joined the faculty at Weizmann in Israel. He set out to take an old anti-cancer drug, one that's still used, methotrexate, and ask, since methotrexate resistance was demonstrated by Shimke and Alt originally to be by amplification of the dihydrofolate reductase gene. So the question is, if you do that and you look for DHFR amplification, methotrexate resistance, what do you get? And here's one such cell example where you can see where we're scoring for chromosome 5, the DHFR gene, and of course DNA in blue. And what you see is, my gosh, look at all the ecDNA. You shattered an original chromosome 5 and produced multiple copies of this ecDNA. And indeed, again, it's easy to rationalize if you have a chromosome breakage to make an acentric piece. Here we are, the acentric piece. It will be encapsulated in the earliest next interphase, encapsulated into its own micronucleus, where it'll undergo slow DNA replication and shattering by the next mitosis, and then religation in these little circles, which can ultimately be amplified. And they can be amplified by multiple means, including by their missegregation, putting many more copies into one daughter cell than into the other. Okay, so is it that shattering, that methotrexate dependent shattering and production of a parent ecDNA, is it really true? Again, with Peter Campbell, we used whole genome sequencing to realize, yes, indeed, in this particular example, the ecDNA contains 5 different pieces of the original chromosome 5 onto which DHFR resides. And if you, at the whole genome sequence level, yes, indeed, they were chromothriptically reassembled in the wrong order to produce a 2 megabase 5-piece ecDNA with all 5 pieces coming from chromosome 5, but not necessarily, not all the adjacent pieces, but of course, containing the DHFR gene. Okay, so we know that we can, by making an acentric piece, chromothripsis, you can generate ecDNA and indeed we validated the predictions from the DNA sequencing that we had pieces that were not adjacent to each other in the original chromosome, piece 1 and piece 4, by a dual in situ hybridization. Yeah, we verified the structure of the ecDNA with these 5 pieces ligated in the wrong order. Okay, so now one criticism could be, of course, that's a cell culture example. Is this the kind of amplification, shattering and amplification that drives anti-cancer drug resistance and cancer genome evolution in real cancers? And we partnered with Rona Yeager at Sloan Kettering, who had examples from patients whose tumors were driven by expression of the BRAF kinase. Well, a specific mutation in the BRAF kinase, to which an inhibitor, a very highly selective inhibitor of that particular mutation, has been generated and is in use as a targeted anti-therapy. And in patients who had unhappily failed the initial therapy and had ultimately become resistant to the BRAF inhibitor, when we looked at those, a simple inspection of those shows that, oh my gosh, here you go. In the lymph node biopsy, you see the ecDNA, and you see with this fluorescence in situ hybridization, lots of ecDNAs, actually in varying numbers in the various cells. But with whole genome sequence, again with Peter Campbell, what we identified is, yes, there's a complex ecDNA formed with multiple pieces containing, in every example, containing the BRAF gene, so that the resistance to the inhibitor is by just amplifying the original target gene. So indeed, we will argue that this is the mechanism of genome amplification in cancer using chromothripsis, ecDNA formation to drive, in this case, resistance to a very targeted anti-tumor drug. So if that's the case, can chromothripsis be targeted to prevent drug resistance? That was one of the 6 challenges in the original CRUK and NCI challenge from 2020. And so we set out to try to do that. Well, what if we now look for this methotrexate generation of methotrexate resistance by this genome shattering and religation, and ask, well, what if we use inhibitors of non-homologous end joining? So when you have double-stranded DNA breaks, you got to put them back together by some mechanism. The most well-known mechanism is non-homologous end joining. You could also, though, inhibit DNA repair with an inhibitor like PARP inhibitors that are now broadly in clinical use. And when you do that, if you add either inhibitors of this non-homologous end joining or inhibitors of the DNA repair pathways using DNA PK inhibitors or PARP inhibitors, yes, you can really drive that down, drive down the ability to generate resistant cells and to generate these ecDNAs. And we propose from this that a combination therapy, which included the use of PARP inhibitors and an NHEJ inhibitor, really ought to be able to diminish acquisition of drug resistance, for example, in that BRAF inhibitor that I indicated earlier. Okay, so would increasing drug dosage overcome ecDNA-driven resistance to it? And we tested that in the DHFR model by increasing the level of methotrexate and asking what happened. And the answer is perhaps no surprise at all, but the answer is, well, you generate more ecDNA. So you go from about 20 per cell, you now got about 60 per cell, and they're just perfectly happy. But if you either use sequencing and or use, as in the example on the right here, fluorescence in situ hybridization, you can see very quickly what happened. Here's the in situ hybridization of a single copy intact chromosome 5 with a single copy DHFR gene, and here the duplicated chromosome chromatids after replication. And here is the fluorescence intensity of the newly made ecDNAs, and you can see they're 10 times as intense as the endogenous ones. So they went from an average copy number of 1 copy on average to about 3 copies of the DHFR gene per ecDNA. And in the example I'm showing you, it's an outlier where it got up to 10 copies. So we argue that yes, and you might say, well, it's no surprise that you add more ecDNAs and you add more copies of the resistance gene on each cDNA, so there is an evolution of that cancer genome. Okay, and moreover, of course, how do you get that evolution? Well, how do you segregate these ecDNAs, these little circles? And Jeff Wall and his colleagues at the Salk had demonstrated in the 80s and early 90s that they like to tether 2 chromosomes. And we demonstrated working with Bing Ren that they really like to tether near chromosome ends, but not all of them do that. And so they're not being passively translocated during mitosis along with a chromosome to which they're kind of tethered, but the untethered ones will not join the bulk mass and will form their own micronuclei. Oh, and when they're in micronuclei, they're substrates for new rounds of chromothripsis. And indeed, that's exactly what happens. So as you can see very quickly, if you start with one of these examples of a simple ecDNA generated by resistance to methotrexate, there it is, 5 or 6 pieces ligated together. We can score it at the nucleotide resolution, but if you select for increased resistance over a 2-week period, look what happened to that ecDNA. It was shattered and reshattered and reshattered and reshattered again. So the evolution of genome evolution is quite easy by this increased drug selection. So we conclude from all of this that we know that if you take a normal chromosome that has a gene whose presence can be selected can provide a growth advantage. That if you break that chromosome with that, or that chromosome gets missegregated into a micronucleus, that it will be shattered by a process of chromothripsis. It will, a proportion of those will go into double minutes or ecDNAs, and indeed you will drive this cycle of evolution of those ecDNAs as they acquire additional copies of the resistance gene, and they amplify in the most resistant cells, amplify the number of ecDNAs. I only briefly touched upon today the alternative route for gene amplification, which was by making these homogeneous staining regions using what's been called breakage fusion bridge cycle, discovered initially by Barbara McClintock, but absolutely that occurs. And we demonstrated, but I don't show today, we demonstrated that these DNAs that become dicentric chromosomes, they get the DNA stuck, especially with the amplified gene in the center, and it gets broken. You get micronuclei, they get shattered by chromothripsis. And so the HSRs are a predecessor of the ecDNA and more widespread gene amplification. So if I come back to where I started with the 2020 Cancer Grand Challenge, the question, one of the 9 challenges was, what is the origin of gene amplification in extrachromosomal DNA in human cancer? And we argued that to the first of the questions, what drives ecDNA formation? Oh, it's chromothripsis. Can we target cancers driven by ecDNA? Yes, with small molecule DNA repair and NHEJ inhibitors. How does ecDNA evolve? By chromothripsis. And can we target ecDNA evolution? Yep, the same small molecule inhibitors. What is the role of ecDNA in cancer formation? Well, at least one role is for gene amplification. Another role will be for inactivation of tumor suppressors, because the fragmentation will ultimately inactivate some suppressors if they are on the chromosome or the chromosome piece that gets fragmented. And can we eliminate ecDNA? Well, that's not really been achieved. Maybe my check marks over the top here. But with small molecule inhibitors of DNA repair and NHEJ inhibitors, I think we can diminish it. And what's left? Well, key mechanistic questions that are left are, so how does a chromosome get shattered? What produces that shattering? And there are ongoing proposals of nucleases that are involved. My own view and from our own work is that the published ones are certainly not sufficient, and they may not be involved at all, at least in many cases. And the final question, really, I think from a basic science perspective, quite of high interest. How do you put all the pieces back together? It's kind of the Humpty Dumpty. When Humpty Dumpty fell off the wall, how do you put 100 pieces back together? And there is today, that's a challenge, a really unmet challenge. And so then lastly, for evolution of genome instability in cancer, this work was begun by a former graduate student, Beth Weaver, now on the faculty at University of Wisconsin. But the work that I really highlight today began with Peter Lee, now independent at UT Southwestern, who built the cells in which we can selectively missegregate the Y chromosome. But in particular, Ofer Shoshani just joined Weitzman, who demonstrated the drug resistance, all of the drug resistance experiments that I described today, and a partnership with Peter Campbell of the Sanger Center for the whole genome sequencing. Well, thanks very much, Tom. Thanks for the opportunity.
Well, thank you very much for the very stimulating lecture. I think that it's it's a thing that always bothers us in terms of looking at the heterogeneity of these different lesions and how complex the karyotype and chromosomal rearrangements are in cancer. And you got at the aspect of inhibitors of DNA repair or non-homologous end joining as being able to treat this, but how effective are they when the chromosomes are already altered in such a way? It may become very difficult.
Yeah, well, you're certainly right about that, but I do think you might, and you could argue that if you can't get rid of them altogether, that even one left will ultimately amplify its way back. And that's actually a quite sensible criticism, which I fully accept. But you can certainly diminish their number, and we would argue that, for example, if I go back to the BRAF inhibitor examples there, where obviously the on-target molecular therapy is to inactivate the kinase that's driving the tumor. Okay, great. So maybe there it's that if you could try to suppress the ability to form these ecDNAs during the original BRAF kinase inhibitor, that maybe we really could diminish the number of resistant patients that emerged.
One of the interesting things is sometimes treatment with these kinase inhibitors can result in mutations within the kinase gene itself. However, it's kind of curious. Sometimes you find that the allelic frequency of the mutation is less than what you would expect if you had 100% of the cells expressing this. But could this be also due to gene amplification?
I absolutely, I would very much think so. Yes, absolutely. And it's easy to not find the amplifications. For example, an ecDNA, it's easy not to see it. It's harder to look. It's actually quite easy to, if you're looking, it's easy to find. But if you're not looking, it's easy to miss. And so I think yes, absolutely that's likely. And in all the examples that we got from Sloan Kettering, while I appreciate that there are all additional kinase mutations, the ones we saw, the resistance was driven by gene amplification.
That's very exciting. I think that in the interest of time, we're probably going to have to move on, but I thank you very much for that stimulating lecture. And it's great to see that the work is proceeding as it is. So thank you very much.
Yeah, my pleasure. Thanks.